Hello everyone and welcome back to Neuroscience Methods 101. Today we are going to talk about event-related potentials. When the neurons in our brain are active, they are often active together in synchrony. This synchronized activity leads to rhythmic patterns, which result in a wave-like signal. These brain waves, or neural oscillations, can be picked up by various machines, including electroencephalography and magnetoencephalography, abbreviated as EEG and MEG, and we discussed those techniques in previous videos. Now, brain oscillations are happening all the time and can reflect various brain states, such as sleepiness or alertness. But rather than looking at these signals over a long time, at random moments, it is also very interesting to time these signals to a particular action. In other words, every time when you do a specific thing, we are going to cut out a piece of the brainwave signal. The distinct waveforms that you will be able to pick up in relation to this particular event are referred to as event-related potentials, which are abbreviated as ERPs. Let's take a look at an example. We are going to show a participant the same stimulus for a while, let's say green dots. Now, if we eventually present a different stimulus, like a red dot, that would stand out. This outstanding signal would lead somewhat to a surprise, as it is different from the general pattern. Such a surprise response can be seen in our brain oscillations when we analyze the ERPs to that event. Specifically, we can observe two components, the mismatch negativity and the P300. The more surprising an event is, the larger these two potentials will be. But ERPs can be observed in a large variety of situations. For example, when seeing a face, a so-called N170 component can be observed. When listening to a faulty sentence, the so-called N400 and P600 components can be observed, which encode the semantics and syntax of language. And when you have to suppress a certain response, for example, not push a button, the so-called N200 component can be observed. And these are just a few of many more examples. Now, to see clear ERP patterns, a single trial is often not enough, and it can only be observed when a number of trials are averaged. Although some ERPs have special names, many of them include the letter N or P, followed by a number. The N stands for a negative peak, and the P stands for a positive peak. The number after N or P indicates when the waveform occurs. A small number, like 1, 2, or 3, suggests it is the first, second, or third positive or negative peak in a particular time window. A large number, like 100, 200, or 300, suggests that the positive or negative peak is observed after 100, 200, or 300 milliseconds on average. It is also important to consider where an event-related potential comes from, since the same names can be used for different processes. For example, components N1 and P1 that originate from the visual cortex reflect basic visual processes. But the same nomenclature, N1 and P1, is also used for ERPs that come from the auditory cortex, and there they reflect basic auditory processing. So when discussing ERPs, the abbreviation is not enough. You also have to specify where the ERP comes from or to which process it is related. Differences in ERP components can indicate differences in the processes they reflect. Deviating ERPs can therefore be an indication of motor, sensory, or cognitive deficits. Therefore, ERP abnormalities are investigated in clinical studies of various psychiatric and neurological disorders. Anyway, that's it. We hope you enjoyed this episode, and we hope you learned something new. If you did, consider liking this video. And we hope to see you the next time. Bye bye!